Now, we are coming to the 31st and last project of our National Contest for Young Scientists for today. This project will be presented by Paula from the International School of Luxembourg, and her project is called An Investigation in the Effect of Ibuprofen on Chlorella pyrenoidosa Growth. Using Chlorella as a model, she studied the effect of a range of ibuprofen concentrations deriving from waste and effluent water on microalgae as a result of improper disposal over 10 days. It was found that the concentrations stimulated growth, but there was no great difference between the concentrations. Possible reasons for the stimulation of growth are discussed, along with an evaluation of the limitations of the methodology and possible improvements. Good luck, Paula.
Humans' impact on the environment. It's a very active field of research, but I believe that we need to take a closer look at just how our healthcare system is a part of this effect. My name is Paula van de Paverd, and today I will be discussing my exploration into how ibuprofen concentration affects the growth of Chlorella pyrenoidosa in order to investigate the effect of pharmaceutical pollution in aquatic ecosystems. After some preliminary research, I found that due to various factors like rise in population, increased urbanization, and an aging population, the use of pharmaceuticals is rising. There are a few possible ways that the pharmaceuticals can enter waterways, with the main ones being through patient excretion, livestock treatment, which then enters through excretion, and direct release into the wastewater system during the manufacturing process. These drugs are being found at various concentrations in sewage water, surface water, and even groundwater. Now, investigating every pharmaceutical in the world and its effect would have been a little out of my reach. So I looked into the most prevalent medicines in waterways to figure out which specific drug I could investigate. According to a report by the EEB, diclofenac is the most prevalent drug worldwide, with detection in 50 countries, groundwater, surface water, or drinking water. Next is carbodiazepam, with detection in 48 countries, and then ibuprofen in 47. Out of these three, ibuprofen was the only one I could get without a prescription, so it's the one that I used for my experimentation. I was already aware of some of the possible effects that pharmaceutical pollution could have on aquatic environments. However, I had limitations when it came to experimentation. There was ethical limits to do with the use of complex organisms, as well as physical limitations, as I only had access to a high school laboratory. Because of this, I was restricted to simpler organisms. However, I realized that this could be to my advantage. I wanted to construct an experiment that was as realistic as possible, so I knew that I had to use an organism found in an aquatic ecosystem. I also thought that researching the effect of ibuprofen on a producer would give the most relevant information, as it would have ramifications throughout the entire food web. With this reasoning, I decided to use microalgae in my study, as they are the primary producers in almost all freshwater ecosystems. Chlorella pyrenoidosa became my microalgae of choice for two reasons. Firstly, it is often used as a model organism for microalgae due to its high growth rate, and secondly, I already had it available as we had a frozen sample at school. Using Chlorella also allowed me to gain some insight into how ibuprofen could impact higher plants, as it had a similar structure. My experimental design consisted of two parts: the culture and the testing. The culture was relatively simple. I used a nutrient medium and a pump as an aeration method and grew 2 liters of Chlorella pyrenoidosa culture. For my experimental design, I first had to make a range of ibuprofen concentrations. To do so, I looked into literature about concentrations detected in various water systems. This ensured that the data stayed relevant to real levels of pollution. On this slide, I have put some of the ranges of ibuprofen concentrations detected in various water systems. It is, however, relevant to note that some of these concentrations also include the metabolites of ibuprofen, something which I will discuss briefly later. From this information, I decided to make a continuous range of ibuprofen from 0.1 to 0.5 micrograms per liter with 0.1 microgram per liter intervals, as well as 2 higher concentrations of 1 microgram per liter and 10 micrograms per liter to have some information about the slightly higher concentrations which were also detected. I then mixed these concentrations with some of the chlorella and aerated them on orbital shakers over a period of 10 days, taking a reading each day. I measured the growth change of chlorella by percent transmission with a colorimeter set to 490 nanometers. For my experiment, percent transmission and growth of chlorella is inversely related, meaning the higher the percent transmission, the lower the chlorella concentration and therefore growth. From my experiment, I found that all the concentrations containing at least some ibuprofen had a lower percent transmission than the control with no ibuprofen, meaning that they had more growth. Notably, the 0.4 microgram per liter concentration had the highest growth. However, apart from this, the concentrations were all relatively similar, as can be seen from the straight line of best fit on the slide. An article on this subject posited that certain biodegradable medicines could act as carbon sources for the algae and stimulate their growth. However, I cannot necessarily draw this conclusion from my data. My experiment also contradicts another article that I found on the effect of ibuprofen on chlorella, which stated that it inhibited the growth by 50%. However, this experiment was undertaken with concentrations ranging from 71 to 89 milligrams per liter, which is quite a lot higher than the concentrations I used. Inhibiting growth would have reduced the nutrient source in the aquatic ecosystem, and therefore would have affected the higher trophic levels. However, my results would have a different impact. One possible effect on the other trophic levels would be bioaccumulation, which is a gradual increase in contaminant concentration in aquatic organisms due to uptake through the environment. Eventually, this increase in concentrations could lead to toxicity in higher organisms, which could have negative effects. The mode of action of ibuprofen is to inhibit an enzyme which treat inflammation and pain. This would likely not have an effect on microalgae. However, if it accumulates in other organisms such as fish, it could interfere with their metabolism. As stated before, the concentrations in effluent water also included metabolites of ibuprofen. One article proposed that the toxicity of ibuprofen metabolites was actually higher than in its pure form. This could worsen the effect of the bioaccumulation. Moreover, since the results showed slight stimulation of chlorella growth when exposed to ibuprofen, the possibility of eutrophication should not be ruled out. This is when there is a large growth of algae which covers a body of water and restricts sunlight from getting to the bottom. This means that photoautotrophs in the water will die and can lead to a chain reaction affecting the entire ecosystem. Now, there is one main limitation to my experiment. As it was at school, I cannot monitor it during the weekend. Therefore, it is possible that some of the controlled variables were not controlled during this time period. Due to this, I believe that the experiment should be undertaken again in conditions that can be monitored constantly to ensure that the results are replicable and therefore reliable. I also believe it would be valuable to investigate other methodologies to look at the effect of medicine on chlorella. For example, change in dry biomass would be another methodology to measure growth. Looking into oxidative stress on the cells could be used as a biomarker to how the toxicity of the ibuprofen is affecting the cells, and looking at the cells under an electron microscope would show if there has been any structural damages to the cell. It would also be valuable to use paper chromatography to determine the composition of the metabolites, as it would allow for a more accurate understanding of what substance is causing the effects. By doing these follow-up experimentations, we would broaden the applications of the study. Determining exactly which metabolites are present, to what degree, and how they affect chlorella as a model for microalgae allows water treatment plants to be more effective in the removal of the pollutant. Moreover, better understanding of how our medicines affect the environment gives us incentive to look at our drug use and evaluate any changes which may be necessary to diminish our effect on the environment. Thank you.